Hi, everyone, and welcome to week 2. So, this week we're starting something new with the recorded lecture and embedded questions within the lecture. Now, I know this week we're reading about and talking about a lot more than genes and heredity, but just from past experiences, I've noticed that this usually is the section where students need the most support and have the most questions about. So what I'll be doing is focusing our lecture today on genetic transmission and heredity. So let's get started. So let's talk about genetic code to begin with. You're going to learn that every cell in the typical human body, except for the sex cells, has 23 pairs of chromosomes, which means that if you get 23 chromosomes from your mother, 23 chromosomes from your father, you have 46 chromosomes all together. That's what it means by 23 pairs. One set from each of your biological parents. Now, how do these cells divide? How do we come up with these different sets of chromosomes? There's two different processes that you're going to read about. There's both meiosis and there's mitosis. And a good way that I try to remember these two different processes is that meiosis is responsible for the splitting of sex cells, or what we'll refer to as gamete cells. All other cells divide according to a process called mitosis. So I look at the T in mitosis and think autosomes are all other cells that are not sex cells, whereas meiosis has that extra S, no T, and so I think sex cells, meiosis. So just my little mnemonic device. If it helps you, great. If not, just ignore it. But that's just how I remember the difference between meiosis and mitosis. So going back, if we're looking at how sex cells divide, we're also seeing that when they divide, each different division is coming out with its own set of information. So each sex cell ends up with only 23 chromosomes, because remember, that sex cell is going to combine with either a male sex cell, or if we're talking male sex cell, it's going to combine with the female sex cell, and together they're going to make the 46 chromosomes that make up a human being. So we're saying sex cell, we're saying gamete. What are we really talking about here? Sperm and ovum. That's what we're talking about. So when we say sex cell, we're saying a sperm cell has 23 chromosomes, and an egg cell or an ova cell has 23 chromosomes. When they combine during fertilization, that's when they pair up, and now you have 46 chromosomes total. Okay, so that's meiosis. Mitosis, on the other hand, is how every other cell in the human body divides. And they're going to divide in half over and over and over again. And what's neat about mitosis and with autosomes is every single cell has an identical replication of your DNA. And your DNA is that information of what makes you you. And we're going to talk a little bit more about that in a second. But every single cell in your body has your DNA present. Fun fact, the only cell that doesn't have your DNA presence is actually the red blood cell, and which is interesting because if you've ever watched any kind of crime show, you would think that the red blood cell definitely has your DNA, but it's actually the only one that does not have a replication of your DNA. Okay, so here's a visual example of what happens. So you'll see these parent cells, and then you'll see the replication. And up here, you're going to see mitosis, which is how autosomes divide. And then down here, you're going to see meiosis, which is how sex cells divide. And by sex cells, again, we're talking about sperm and ova. Here is a really cool map of all these chromosomes, and the specific word for this is called a karyotype. And so as you can see in this karyotype, you're actually seeing the combination of chromosomes here, and what geneticists do is align these different chromosomal pairings into a pattern of pairings. So you're going to see 22 pairs of chromosomes, and then what's unique here is you're going to have a pair of sex chromosomes. So that 23rd pair are your sex chromosomes. So we're going to look and see what that means for each individual. So what determines sex? All ovum, so all of the female gametes or female sex cells are X cells, so are X chromosomes. So every single ovum is an X cell, X chromosome. Sperm, on the other hand, can carry either X or Y. So what does that mean for who determines sex or what determines sex? It's actually the male gamete that determines if a fetus is going to be male or female, not the woman or the egg cell that determines that, because all ovum, again, are X. So if an ova is fertilized by an X sperm, now you have an X ova and an X sperm, and that means that the baby is going to be a girl. If you have an X ova fertilized by a Y sperm, now you have a baby that will be a boy. There is 1/3 designation of sex at birth, and it's known as intersex. And that means a child could have either an XX or an XY in their chromosomal makeup, but they're presenting their phenotype. There's some presentation of characteristics that are not specific to that identification of girl or boy. For example, I could have an XX combination and be determined girl based on my sex chromosomal pairing, but maybe this girl also has the small resemblance of a penis. So that child, those combinations of characteristics are actually given the determination of intersex at birth. So you can either be designated girl, boy, or intersex at birth. Okay. Now, what is epigenesis? We're going to talk about an epigenetic framework. And what epigenesis is we've learned that it's not just nature and not just nurture that frames who we are and how we develop. And what we know is even with biology, you might have a genetic predisposition, but our environments really do play a role in whether that gene is expressed fully or not. So it's this mechanism that either turns the gene on or off and determines different functions of the body. So epigenetic changes can respond to environmental factors. So what you eat can determine whether these predispositions turn on or off. Your sleep habits, how you take care of yourself, and your stress can also turn genes on or off, and also physical affection. So are you being cared for? Are your needs being met when you're a baby? That can also lead your predispositions to either, again, be turned on or turned off. Epigenetic modifications may be heritable. So if you, for example, I have a predisposition for anxiety and depression. If I'm in an environment that is really stressful, that is high pressure, not a lot of support, the chances of my experiences turning on those genetic predispositions for anxiety and depression are really high. Or on the other hand, if I have a predisposition and my environment teaches me how to really get a hold of how I'm feeling, how to acknowledge how I'm feeling, how to express myself in a healthy way, how to handle my feelings of anxiety, then it might not lead to full-on depression. Another example of this is also drug addiction. So there has been a genetic link for predisposition of drug addiction. Now, if I have that predisposition, let's say both my parents are drug addicts and I am in a different environment as a teen or as an adult, and not really have drugs at my disposal, I'm in an environment where I'm surrounded by people who don't engage in drug usage, the chances of me following into that genetic predisposition are going to be lower than if I were to be constantly exposed to that. So genotype environment correlation. So these are more ways that the biology and the environment interact in order to express different genetic um, phenotypes and genotypes. Okay, so passive correlation. Um, let's get, for example, say that if there's a family that's really into music, they love music, there's music playing all the time in their home, they have instruments everywhere, everyone plays an instrument when they gather as a family, it's music time, music is just embedded in their world. Well, if they have a child who has a genetic predisposition or has that music gene, that child is just naturally and really passively going to be exposed to an environment that supports that gene. So the parents don't really have to try any harder. The child doesn't really have to try any harder to really grow that gene and show that gene of a predisposition to musicality. The second type of genotype environment correlation is called evocative correlation. This is when the child has certain genes, so has the musicality gene, let's say, but the parents have no musicality gene. Let's actually switch over to art because this really kind of plays a role in my own family. So my daughter is very artistic, has a lot of skill when it comes to her art. I, on the other hand, have no art bone or gene in my body. And so I'm not very good at it. But when I saw that she had a skill, I actively or evocatively provided her with an environment that says, here are some paints, here are some canvases, here are different programs that I can enroll you in that will support that gene because I don't have that ability myself. The last one is active genotype environment correlation. So this is if the child has always had this longing for music or longing for art or longing for something that is a gene in their body, but never really acted on it until they grew up as an adult and realized, you know what, I've always wanted to do this. I'm going to actively seek out these opportunities to grow my passion, to grow what I know I'm good at. Okay, so 3 different types of genotype environment correlations. Now, what is a genotype and what is a phenotype? So there's a little mnemonic device I like to use for this as well. So for phenotype, I think of face. What do you see? What are those observable characteristics of a person? What's my hair color? What's my eye color? What's my skin color? If you've ever heard, she has her mother's nose, that's a phenotype. That's something that other people can see. Genotypes, on the other hand, those are the instructions. That's the genetic makeup of a person. And it contains both expressed and unexpressed characteristics, but it's kind of like the underlying roadmap of what's going to be expressed in your phenotype. There's also something called the multifactorial transmission. And this is a really all-encompassing view of what we just talked about, that combination of genetic and environmental factors to produce certain complex traits. For example, we might have the genotype for a darker colored skin, but maybe you never go outside and you never see the sun. And that means that your phenotype might not actually match or have other people see a view of what your genotype says. So that multifactorial transmission is important because it really emphasizes the role the environment plays in that genetic environmental relationship. Okay. More patterns of genetic transmission. So what happens with genes, or they're different types of how that gene is going to be expressed. And these are called alleles. And when a gene is being passed on, it's being passed on through these alleles. And it can be passed on in a couple different ways. So there's something called homozygous transmission. And that's when a child it receives identical alleles for that trait. So they receive information that says, this is the same copy of what this is going to look like. This is another copy of the exact same thing of this is what it's going to look like. Heterozygous transmission, on the other hand, is when you have two different alleles. Let's say one of the alleles is being tall and one of the alleles is being short. And you, depending on if that's a dominant or recessive inheritance, it'll determine whether you're tall or you're short, okay? So let's talk about dominant inheritance. What are some characteristics that are a product of dominant inheritance? One of these is dimples, actually. So in order for a child to have dimples, let's say everyone in the family has dimples. The chances of that family passing on a dominant dimple gene is pretty high. So even if that child gets one dominant allele and one recessive allele from another parent who doesn't have those dimples, they only need one copy of that dominant allele in order for that trait to show up. So let's say mom has dimples, dad has no dimples. Mom passes on a dominant dimple allele. Dad passes on a recessive dimple allele, meaning no dimples. And when that child is born, they have one dominant allele from the mother and therefore have dimples. Here's an interesting fact with recessive inheritance. I want you to look at your hand, and just one of them, and if you have 5 fingers, you actually have the product of recessive inheritance. Having 5 fingers is actually a recessive gene, a recessive trait. Having 6 fingers is the dominant trait, which means if you have more than 5 fingers on one hand, it is possible that you had one of your parents pass on the dominant allele for 6 fingers or more than 5 fingers on one hand. But if you have 5 fingers on each hand, chances are you got a recessive allele from your mother, a recessive allele from your father for 5 fingers, and therefore have 5 fingers on each hand. There's also something called polygenic inheritance. And this is when multiple genes, but at different sites of the chromosome, affect a complex trait. I like to use skin color again as an example of polygenic inheritance because it really is determined by so many different genes, and it could be on chromosome number 3, chromosome number 7, chromosome 21, let's say, and all of those genes play a part in what the final skin color is going to be, how that skin color responds to sun, how that skin color burns, how that skin color either displays freckles or not. They all play a role in the phenotype of that characteristic. Okay. Let's look at some genetic and chromosomal abnormalities that can occur as well. So again, a dominant trait is examples of dominant traits are achondroplasia and Huntington's disease. In order for a child to be diagnosed with one of these, it would mean that they had one dominant allele from one of their parents. They only needed one. So if they had let's say their mother had they passed on a dominant allele, but their father didn't, because they have the one dominant allele from their mother, they had the chance of having dwarfism or achondroplasia. The same with Huntington's disease, that same process. And I'm going to show you a visual in a second. There's also something called recessive traits and recessive trait conditions. So Tay-Sachs is an example of a child having the recessive traits that end up being considered an abnormality. So with Tay-Sachs, the child gets the recessive genetic mutation from one parent and then receives that same genetic information from the other parent. So they have to have those matching or identical genetic transmissions from each parent. The chances, because those recessive traits are not very common, when they do produce these abnormalities, the chances of survival are typically lower than children who are born with dominant traits. Tay-Sachs disease is probably one of the most devastating conditions I've seen. It's very popular in the Ashkenazi Jewish community, and typically children are developing just as they should be, meeting milestones, and maybe about 9 months old, all of a sudden you see all that progression stop and then decline severely. Typically children with Tay-Sachs disease do not survive past their 6th birthday, and so it's a very devastating condition for their families to be a part of. Another example of a recessive trait is sickle cell anemia. And sickle cell anemia can be a product of incomplete dominance as well, which means that the child has both recessive traits, but one of the alleles might not have all the information that it needs. So that's what it means by incomplete. And so children with incomplete dominance means that they won't, differ in severity, so they might not have it as severe, they might not present physically with the condition. It's going to be a different type of presentation when the transmission is incomplete. Okay. All right, let's talk about sex-linked inheritance. So if it's sex-linked, chances are that it's being passed down on either the X of the mother or the X that is passed from the father, because remember, all ova are X, but the father can either transmit an X or a Y sperm. And so typically this pattern is inherited from the mother and transmitted differently to her male and female offspring. So I want you to think about, why would it show up differently in baby boys compared to baby girls? What do baby girls have that baby boys do not? An extra X chromosome. Remember, girls have an XX chromosomal pairing, whereas boys have an XY chromosomal pairing. So if females get an unhealthy X from their mother, they have another healthy X that sometimes can cancel out the genetic information from the unhealthy X. Whereas males don't have that extra information from another X. Instead, they have a Y that doesn't cancel out the same way. Okay. Here's some of those examples. So let's say the mother is a carrier, the father is not. They have one affected X and one non-affected X. So you'll see that the affected X will go to 2 children, but they're also getting information from their father. So if that child gets a Y from their father, chances are that that child will be affected by that condition. Whereas if the child were to get another X, they would still have an affected X, but they wouldn't present with that diagnosis. Instead, that female would now be a carrier of that condition. So having the same chances as their mother of passing on that condition to a future generation. Examples of sex-linked disorders are Fragile X syndrome and Duchenne's muscular dystrophy. So each of these is more typically found in boys, but it's not, does not mean that girls aren't affected at all. We do see girls with Fragile X syndrome and with Duchenne's muscular dystrophy. They just make up a much smaller percentage of children who are diagnosed with these conditions. Let's talk about an autosomal chromosomal abnormality, and we're going to talk about one specifically, and this is a karyotype example of a child with an autosomal chromosomal abnormality. And what that means is this child either has too many or too little of an autosomal chromosome, which means anything but the 23rd pairing. So any other chromosome either has too many or too little. So if we look here and look down into 21, we see that instead of 2 chromosomes, there are 3 there. And if you don't know yet by us talking about it, this is called trisomy 21, more typically known as Down syndrome. But this is the karyotype of a child with Down syndrome. You can see the 3 chromosomes here instead of 2. Okay, these are just some images of children with Down syndrome that I wanted you to see. If working with children with unique needs and talking about how to support children with unique needs and their families is something that you are interested in, I really strongly advise you to take the Child Development 163 class, which is the curriculum and strategies for children with special needs. Another example we're going to talk about are sex-linked chromosomal abnormalities and how they actually occur. So it's an error in the division process. So we remember how sex-linked and gametes split, right, through meiosis. And so through meiosis, there's an error. And so there's either an extra or a missing sex chromosome. So there's something called triple X syndrome. There's also something called Klinefelter syndrome, which means the child has 2 X's and a Y in their sex chromosome. And then there's something called Turner syndrome, where the child has an X and an O instead of an XX or an XY. So children with Klinefelter syndrome, where you get 2 X's and a Y, that child is considered male because of that Y chromosome. But what we're going to see is atypical development of those male characteristics. With triple X syndrome, that child is determined as a female, but the same thing, we're going to see some irregularities in some of the characteristics of what it means to be female. Same thing with Turner syndrome. When there's an X but an O, that child is still going to be female, but going to struggle in several different aspects of being female, including infertility. We're going to see underdeveloped sex organs, even atypical growth patterns, and impaired spatial abilities. Down syndrome, again, is another example of a chromosomal disorder. Because it is passed down from the mother, you can get an extra chromosome instead of the 2 that are supposed to be there. Okay. Here's some images and some features of both Klinefelter syndrome and Turner syndrome. So this is Klinefelter syndrome, this is Turner syndrome, just in case you were curious about what are typical characteristics of each condition. Okay. So that's really what I wanted to talk to you about today. I know this is typically the section, again, where students have the most questions, where it gets the most confusing because I know you didn't sign up for a biology class. I'm hoping a lot of this sounds familiar to you, but if it's all brand new, please don't hesitate to reach out and ask questions. I know it can be confusing, but it's important to know about the biology and the foundation of biological transmission so we can see how the environment and biology play a role together in the development of children. So I hope you enjoyed this lecture. I hope you were able to get some questions answered, and I hope this helps you in your learning process. Bye.